That's why it's good.
Okay, so question one here. It's asking about where is the location of an example parasite? Anybody want to volunteer their diagram for being shown up here?
Would it be between any of the two segments?
Okay,
Segment in between two segments?
so what would that look like?
Take A5 and put a line through A4 and A5 and that.
That's what I wanted to see. Show your picture.
Mine is.
Show your picture. Okay. This group, okay, they think that it goes that way. And this is correct. So you're starting here from the top of a stained portion, which is mid-segment,
Bless you.
and moving down towards the end of the non-stained portion. So from here, indentation to indentation, that would be a segment. But this group is correctly drawn from mid-segment to mid-segment is a parasegment. So you could also draw it, you just want to write on their paper.
Yeah.
We went right from here.
Can you see that out? This, right here.
There, down across the top of this stained bit. So now we have one half of a stained bit and one half a non-stained bit. That would be a parasegment. Does that make sense? Okay. So it's the same width as a segment, but offset 50%. Okay. Good job. Okay, the next question we have is in this embryo here, an OTGT mutant. OT stands for Oncofeltus Sphosius, which is the name of the creature, and then GT actually stands for giant, which is the gene that we're looking at here. This is a mutant in B, so which segments or parts of segments are missing? Yeah. MX through LB is gone, and then A1 and A4 are gone. Okay, so there actually were two regions that got deleted in this embryo, and they actually are not right next to each other, but there's two of them.
Would it be wrong to just say A1 through A4? Is that different?
Well, that's only half of what's missing here, right? So A1 to A4 is missing.
Well, no, I know. I'm just saying.
Is that acceptable, rather than saying A1, A2, A3, A4?
Oh, yeah,
Should I
sure.
say A1?
Yeah,
Okay.
Yeah.
yeah, I know what you mean, so
Okay.
that MX and LB they're missing. So based on that, how should we classify this? Cytoplasmic polarity, which is one of the like maternal products, might be missing. Gap gene, parallel gene, cycle polarity. What fits the best? Gap gene. Why? Missing a whole series of consecutive segments, right? So instead of one piece of each segment, like cycle polarity, or one piece of every other segment, like parallel, this time we have some consecutive regions of the embryo that are adjacent that are now gone. And it does happen to be two of them. Some of the gap genes might have only one missing region, and this one has two missing regions. And some of the gap genes happen to be expressed in two broad places instead of one. Okay, very good. So next one here. If we look at the picture in A, where do we think that this B gene, this mutant gene, would have been expressed in the wild type embryo? Sorry? Right. It would be expressed in the same chunks that are missing in B. So we're expecting it should be expressed in the MX and LB segments, and then the A1, A2, A3, A4 segments should be expressing this gene. Okay, that's correct. All right, then we get, all right, we have here in B a nice mutant for the OTG2 gene, but if we didn't have one, if we knew the existence of this gene as a wild type copy in the genome, we knew it's expressed somewhere interesting in the embryo like this, but we don't have a mutant, and we still want to study it. What could we do? Yes?
That one where, basically like the example in the lecture today where we inserted the lacquer one in the embryo and then dyed them.
Okay.
That way you can see it dyed in the missing segments.
Okay. So this is a proposal that we can take a reporter gene like LAC-Z, and we could hook it up to the reporter of this gene here, and we could study where the gene is expressed. So we find out exactly when and exactly where it would be expressed. That'd be a nice experiment to do.
You could also like make your own mutant by blocking the production of that gene,
Okay.
seeing what results in that.
How could we block it?
Well, that's a good idea.
Single straight.
I mean, you could like put UV on it, but I don't know if that would destroy everything or specifically what you're looking for. You could like target it with antibodies that like methylated it somehow so that those.
Okay,
specific genes
we're on the
would be
right track here,
expressed.
so we're thinking of the
Anti-sense
idea.
mRNA.
Yeah,
Single
that's
straight.
right, yeah. Yeah, so it's kind of two ideas here. One is we do need to block it, and you're absolutely right on for that. And then the question is how. So what we really have here is a reverse genetic strategy that we want to carry out, right? We have a gene of interest. We want to get rid of the thing, and we need a mechanism to do so. So antisense would be a good way to do it. Any other possible things that might work here? Think back to our techniques lecture where we talked about some options for different animals. Sorry? A movement experiment? So that's a little bit different than a block-it experiment. What I'm asking right now is just for methods to block it. Yeah.
Oh, methods to block.
Okay, well, what were you thinking?
We were thinking about antibodies, specific ones, like C, because we didn't
Okay,
know what
you were thinking an antibody. So if you had an antibody to this gene and you looked at to see where the protein expression was, that would give you some information. Would that fall under show it, block it, or move it? Show it. Okay. So what has been proposed here is that we should make a mutant, or we should somehow block the expression of the gene.
Well, I have two ideas. One is that you could, like in those examples where we cut it out from the embryo, the original gene, or the
Okay.
original maternal product, if that's only seen as maternal product. And then the second one I was going to suggest is that you could do something to the protein itself, which would be another pseudo blocking technique.
Okay, so a couple ideas of maybe somehow, if it was discretely localized, we could suck the gene product out of that embryo. I don't know of any actual real-life examples where that's done, because it's hard to make sure you get all of it, and,
Would you just stop transcription of some?
okay, stop the transcription. How could we do that? we talked about a couple different methods, yeah.
Well, if it was, if it's counterbalanced by some of the other gene products that we had earlier, you could insert more of the other gene products.
Okay, so you're thinking of maybe a range of mutant background where the endogenous gene would become super repressed and never turn on? Good idea in principle, hard to do in practicality. How about something we actually talked about in class? Just a general method. Somebody back here had an idea. You're in the green shirt there. Was that you who had an idea?
I'm sorry?
For how to block the transcription of a gene? Somebody back there was raising their hand. I thought it was you.
Oh, we're just basically going to propose doing like the in situ hybridization,
Okay,
basically.
okay. Yeah,
Aren't transcription factors used to suppress?
yes, transcription factors are used to suppress.
So you could use a morpholino sequence.
There you
to
go,
block this
morpholino.
for that.
That would be one of the methods we could use. So we could use antisense, we could use morpholino. If this were a mouse, what would we do?
Knockout.
We'd make a knockout mouse. Because you can do that in mice, but we can't really use some kind of organisms. So it kind of depends on what the organism is, what technique we would use to employ the genetics. So now what I'm hearing from you guys are all nice, thinking back to different lectures and different things we've talked about. But I just want to make draw one distinction is that for showing information, we're going to use techniques like in situ hybridization, antibodies, and so forth. And that's not really disrupting the system at all. It is still teaching us something about the gene, but it's not teaching us about the function of the gene. It's only teaching us about where you're going to find this gene and setting up a hypothesis. So if you really want to look at function, you need to do a block it or a move it type experiment, and there are some techniques that have been worked out for each organism. And then there are some creative ideas like some of you have had that people might actually try if they got in a pinch. But if you have a nice established method, you can easily use that. Okay, all right, so that's our 5 questions. And if we want to pass these over to one side here, we can collect them for some points. And then meanwhile, if you want to get ready for the review part, if any questions. Andrea, if you want to walk along and collect them.
Oh, very nice. Just block it. Yeah. Is that not, block it is, usually, or is it also?
Okay, there's been a proposal on the floor that we could use RNAi to knock down this gene. And that also would be a very nice method to do so. And a question about whether that would constitute a complete knockdown of the gene. And so these techniques like morpholino or antisense injection or RNAi, all of these, of course, are targeted at removing RNA species from the cell rather than changing the DNA sequence. And so there's going to be some positives and negatives to that technique. The positive is it's quite easy to do it. You can just, as long as you know the sequence, you can quickly make the reagent, inject it into the embryo, and wait and see. On the other hand, it's hard to make sure there's no off-target effects, so some non-specific side effects that you might get just from loading up embryos with buckets full of this antisense RNA. And it's hard to be sure that you're getting 100% full knockdown removal of any protein product from your gene of interest. You know, you can usually get pretty good, enough to see a phenotype and enough to answer your question about what is the wild type function of this gene usually doing. Okay, so for now, for the rest of the period here, I want to have a review session. And hopefully you've had a chance to go ahead and look at the spring exam that's posted online. And if you haven't done that, I would recommend doing so because it will help you to register about what the scope of what I'm going to ask is. Because we've gone through tons of stuff and really quickly. So if tomorrow you're gonna be feeling kind of overwhelmed. And it would be nice to have more of a sense of what are we going to be asking on the exam. So one area to focus on is that each of your study guides has these vocabulary terms listed on that. And I like to tell people that you can already enhance your grade just by making sure that you know all those terms completely in cold. Now, some of them are more important and some of them are less important, so you can take that into account. But if it's not on that list as a gene product or a vocabulary term, then I won't say it's not important, but you won't really be tested on it for exam. And then another thing is that in each class that we've gone through, there's been some major new concepts, some new ideas, some things that talk about mechanism for how an embryo is trying to accomplish a particular goal. And I've tried to actually list some of those as major concepts for the lecture. So when you're studying, you could use that as a tool. You could say, okay, here's a concept that she wrote down. I didn't make this comprehensive, so you may want to add some, but I threw a few in there. And so you could say, here's a concept. What does this mean? Can I articulate this? Could I write a paragraph about it? Could I explain it to my roommate? Just to make sure that you're really on track with all the different ideas. And some of them may be very simple, intuitive, others like maternal inheritance. Maybe you want to go back and kind of see if you really know what that is or not. Then the questions on the study guides are very open-ended, so again, you could use those to really, just as a starting point to branch out to other things. But the idea behind that is that when we look at how people learn and how you can get the most efficient use out of your study time, you only get a certain amount of feedback and understanding from reviewing your notes and re-looking at all the material and maybe reading the book. So what you really want to do is to get in there and wrestle with that in a context that makes you think more deeply. And so these questions are to try to give you a sort of different scenario and ask you what happens here. And if you do that kind of thing, you tend to remember it and understand it a lot better. So with that being said, we have some time now to go through any questions that you might have from any of the lectures. You can ask just random questions of your own. You can ask study guide questions. You can ask questions from the old exam, whatever you want. And what I usually do is try to start kind of at lecture 1 and go through one by one just so that we can have a group of questions all on the same topic and then move on from there. So, lecture 1 was kind of our introduction lecture. Are there any questions on lecture 1? So, let's see here. Yes.
I think one of your study questions for now was like difference between determined and differentiated.
Okay.
And from one of your slides, you just basically say like determined is when they're already committed. And then after they're committed, then they differentiate. That's what you're talking about there. Is that two different? Or are you talking more like, in some species, cells right away from the beginning are going to be a certain thing. Whereas like invertebrates, a lot of those cells can change, differentiate.
Okay.
I didn't know which one you're talking about there.
Yeah. So the question about determined versus differentiated. And you're commenting here that in different organisms, they get to that stage of being determined either really rapidly or they take some time to kind of figure it out. And that question is really more asking about the meaning of those two words and how they mean distinct things. So somebody like to define what it means to be determined for a cell? Yeah.
Committed to one particular developmental stage.
Excellent. Committed to one particular developmental cell fate. Now maybe when you look at that cell in that organism, it may be at that level of commitment and it still looks like a round generic cell. You can't really tell just by looking. So when we say it's actually differentiated in that particular cell fate, what would that look like? How would that be different?
It looks like and has functional properties of that actual cell type.
Right. So it's going to be showing some real functional properties of that cell type. So if it were a neuron, it might be making neurotransmitters and actually receiving electrical inputs. If it were a muscle, it may be actually contracting. If it were just an epithelial cell in the skin, it probably expresses some junctions with its neighbors, maybe some keratin. So, you know, some distinct meaning between those two words. Yeah.
So differentiated is more associated with phenotype?
Differentiated is more associated with phenotype. Although, you know, phenotype is a broad word to mean anything that we can test and see in the organism. So we could maybe test that organism by in situ hybridization and see, yeah, this cell is actually expressing a cardiac marker. In fact, that's one of the ways that we know it's determined, even though it's not beating yet, so it's not actually fully differentiated.
So then on the practice exam, when you say pattern, you mean like a phenotype?
So when I say pattern, I
It
probably
says it has enough patterning to acquire a particular cell fate.
Okay, has enough patterning to acquire a particular cell fate. What do you think the word that would describe that would be? Out of the choice of totipotent, pluripotent, specified, determined, or differentiated.
I've put down differentiated, but it then goes on to say, and is not likely to alter to a different pattern even exposed to new signals. That's also the definition of determined.
Okay,
So that's where I think he's.
so you said it has enough pattern to take on a cell fate, and then the second half was,
And is likely, it is not likely to alter to a different pattern.
okay, and it's not likely to alter to a different pattern. Okay, that would fit with determined
Right.
in my mind, because we're talking about level of commitment.
Right.
So that's more fitting with determined. If we had said it has enough information and it could still change, what would that be? Specified. If I say it has enough information, it's not going to change, and it's expressing a secretory side and some gap junctions, that might be differentiated, because you're really seeing these outcomes of the function. Yeah.
Are you still struggling with the idea that possible differential gene expression?
Okay, struggling with the idea of differential gene expression.
Yeah.
What?
It's like trying to understand, because you talk about, like, it's not necessarily, it's just every cell does it a bit differently. So, the differential part just means that every cell has different mechanisms for essentially.
Right, so the differential part. So this idea with differential gene expression and how that would be achieved by the cells could happen at many different levels. Like the first and most intuitive way that you could think of the cells, two neighbor cells being different, is they express different genes. So they're going to have different mRNAs. They're different in what they transcribe. But that's only like the surface. You could then think of those mRNAs being translated into proteins. Proteins process differently. Proteins could have different kinds of modifications like phosphorylation. And one of the thoughts from this first lecture is that any of those steps along the way till you get to the final functional protein is fair game for doing differently in one cell versus the neighbor. And I think that that could be considered part of the mechanism of differential gene activity. Is there another question somewhere or on this side? Yeah.
Yeah, I wanted to know sort of what you meant specifically by developmental fate. Do you mean like abdomen, thorax, head region, or do you mean like nerve tissue, or do you mean like 3 different types of cells?
So question of what do we mean by developmental fate at the tissue level, at the germ layer level, or at the cell type level? And I think any of those could fit, depending on the context that you're talking about it. Generally, I mean on the cell, the individual cell function level is what I'm thinking when I think of cell fate. But, you know, certainly the germ layers are already, have a different potential for the cell fates that they can give rise to, so it's sort of related. Good, I'm glad you were asking questions. Okay,
So on some of the questions that are pretty open-ended on the test, in terms of what you just answered for her question, like if we did say something along the lines of talking about the germline and you meant in terms of head, thorax, abdomen. If we made an argument for our answer, would we get credit?
Okay, so this is a question about, what if you gave a correct answer but at a different level, so to speak, on an exam? And I'm going to try to make my questions really specific. So first thing is to read the question in detail and see if that can answer, your question about what level or how specific. Secondly, if you're not sure and you want to know, should I go more detailed than this? You can ask me during the exam and I'll say, go more detailed. I won't tell you if this is a right or a wrong answer, obviously, but I can say, I need more specific, or that's specific enough. If I say it's specific enough, I'm not saying if it's right or wrong, so.
Well, just like on the little, in class, and we did the last question, there was a lot of
True.
information that
Right.
you could have stood out at you. And
Absolutely.
Just saying, oh, antisense mRNA, boom, is that good enough? Or should we go into more like,
Yeah.
you could do this and this, and then if that doesn't work, you could do this.
Sometimes it will happen, there'll be more than one correct answer for a test question that I have. I generally try not to have that be the case just so it's more straightforward for grading. But, I'm making these up as I go along, so sometimes I don't pick up every single scenario. And as far as I'm concerned, the right answer is the right answer. So sometimes I will go back and change what we count for credit, depending on what answers people put in the real situation. Okay, let's go on so we don't run out of time. Lecture 2, here is our techniques lecture. Anything for that one?
What's the difference between knocking down and knocking out?
Okay, knocking down and knocking out. There's a distinct meaning here. So in mice, when we knock out a gene, it's usually done. Since you have the ability to basically do anything that you want within the gene, the cleanest experiment is to make a mutation that would fully obliterate any functional gene product being made from that gene. So for example, you could just delete exon 1 that contains this translational start site. If you do that, there's not going to be any protein made from that locus. And then you get a very clean, strong phenotype. On the other hand, when we're talking about these morpholino and these RNAi approaches, we'd like to do the same thing, but we can't fully guarantee that we actually achieve that. And so we generally use the term knockdown to reflect that, yes, we've gotten rid of or reduced the amount of gene product, but maybe there's a little bit left. And therefore, we might be seeing a little bit of a weaker or slight more less dramatic phenotype than could be fully possible if we completely eliminated the gene. So it's a nuance, but it's just reflecting the complete obliteration for knockout, or the probably most of it for knockdown. Yeah.
And morpholino acts the same way as the antisense mRNA. It just attaches to the mRNA and stops.
Yeah, so morpholino versus antisense RNA in principle are the same, but the morpholino is going to be quite short, only 25 base pairs, and it's actually modified on its sugar phosphate backbone to be more stable for the cell so that it'll stick around longer. The first experiments that were done were just RNAs, like full RNAs that were antisense, injected into the embryo, and it worked, it was specific, but oftentimes the RNAs would degrade really quickly, so that wasn't that easy. Okay. All right, let's go on to lecture 3 then. This is the first sea urchin lecture. Talk about some cleavage here and some fertilization for sea urchin. Yes.
Actually, I'm sorry, this is about lecture 2.
Okay.
Can you just review what the tumor-inducing plasmid is and what people use it for?
Okay, the TI plasmid in Agrobacterium. So this is the plant system, and it happens to be that there is this crown gall virus that makes this T1 plasmid. This is found out in the wild And as part of its mechanism, as part of its life history, it's going to insert a portion of the DNA from the TI plasmid into the genome of the plant. So we're literally transferring a bacteria DNA into the plant genome. And so that's a form of a transgene because you're introducing a foreign piece of DNA into the chromosome. Once it's in the chromosome, the plant doesn't know it. It thinks it's a plant gene. So it expresses it, it replicates it, it just treats it like a random piece of DNA. But it's now fully integrated, and any daughter cell would also have that piece of DNA in there. So what researchers have done is just to manipulate that system where we took out the bacterial bits and we put in our gene of interest, like a reporter gene, and now we can get that to transfer over into the genome of plants.
So is this how a lot of like genetically modified crops are made? Or is there a better way to insert genes
so-
in the virus?
These days, there are different kinds of mechanisms to deliver the transgene to the plant or the crop. And it doesn't so much matter what the delivery mechanism is, it matters more what sort of the DNA that you're introducing, so the nature of the transgene. But in principle, it's that same sort of idea. You're making something in bacteria that's interesting to you, and you transfer that over and make it part of the genome. So for crops, they often may have, say, a resistance to an herbicide, and therefore if the plant's resistant to the herbicide, you can spray the herbicide and all the weeds and all the other stuff that doesn't have that gene will die, but your plant now has a special gene and will survive. Yeah,
When you insert this plasmid, does it insert randomly into the plants?
good question. The location of the insert is totally random within the genome for this technology. Okay, sea urchin fertilization. This subject has some terms and it has some orders of events. It has fast block versus slow block. So you want to have clear in your mind all of those things.
There's one term in there, I just couldn't find it, it's polar body.
Oh, polar body. So, yeah, don't worry about polar body. Cross that off. We'll come back to it later.